I presented the results on a multi-institutional study that was conducted within ERIC, the European Research Initiative on CLL, and the subject was the cytogenetic complexity in CLL, clinical biological associations, and the impact on clinical outcome. And the reason we did that is that we know for many years that complex karyotype is a very important prognostic marker in CLL. However, it was superseded by the FISH analysis, and for many years it was at the backstage. Recent reports, however, support the predictive role of cytogenetic complexity in CLL, and a way to do it is by doing classical cytogenetic G-banding analysis. So what we did was to gather clinical biological data on cases that cytogenetic analysis was performed, CLL patients. In total, it was about 3,500 patients from 12 institutes from all over Europe. And what we actually found is that complex karyotype, defined with the old definition by the presence of 3 or more abnormalities, is associated with a very bad clinical outcome and a very short overall survival. However, this was the largest cohort so far analyzed. And in that way, we were able to look within this group of complex karyotype cases. And we ended up showing that it is actually a very heterogeneous group. You can find subgroups that have even favorable clinical outcome. And actually, it is the presence of 5 or more abnormalities, either structural or numerical, that are uniformly associated with a very bad clinical outcome and a very short overall survival, less than 5 years in CLL. We also evaluated the association between cytogenetic complexity and other established markers in CLL, such as aberrations of p53 gene, either deletion or mutation. And we found that even within this group, that is the only group that has a different treatment in CLL, cytogenetic complexity is relevant, and the presence of 5 or more aberrations is even within this group associated with a worse clinical outcome and a survival of about 2.5 years. And the same, the result was the same even when we evaluated the impact of complex karyotype within other subgroups, such as cases carrying unmutated IGHV genes. And the main conclusion is that the identification of complex karyotype is very relevant in CLL. However, we need to be sure about its definition, and we think that we should try to evaluate the complexity in another aspect. Of course, the classical definition from complex karyotype, as we know from other diseases, is the presence of 3 or more aberrations. But it seems that what is clinically relevant in CLL is the presence of 5 or more aberrations, since we have showed that cases with 3 or 4 aberrations have the same overall survival with cases carrying no aberrations at all. This was the main finding of our study.